Please join me in welcoming Rizwan Haq. Dr. Haq earned his PhD from the Ontario Cancer Institute, Princess Margaret Hospital in Toronto, Canada, in the laboratory of Brent Sankey. He completed internal medicine training at Johns Hopkins, the fellowship in oncology at Dana-Farber. He's a physician scientist here at the Farber, specializing in metastatic melanoma and leading efforts to develop the next generation melanoma therapy. Thank you for joining us.
Well, thanks to the organizers for inviting me. I'm going to take a little detour into intrinsic resistance and specifically genomic mechanisms of resistance. And I think this audience knows very well. A important question Dave has mentioned is why, in fact, maybe even the important question for immunotherapy is why some patients respond to treatment and why some patients don't. And this is relevant to almost half of our patients in melanoma have intrinsic resistance. There's this more narrow question, almost a niche question, and that refers to those patients who initially respond to checkpoint inhibitors, but ultimately have progression. And I say that's a niche question because most patients who are treated with checkpoint inhibitors and have response continue to have responses to their checkpoint inhibitors, even when the treatment is discontinued. And so today I'm going to actually be talking about the second question. And you might say, Riz, why are you talking about this niche question when there's this really important big question in front of us? And I would say, you're probably right, but there is a possibility that the answers to the second question will inform the first question. And so today I'm just going to talk about acquired resistance to checkpoint inhibitors. And I think that the sort of seminal initial discovery about intrinsic resistance to checkpoint inhibitors in melanoma was this paper published in the New England Journal of Medicine by Tony Ribas's group. And I'm just going to take you through that paper because I think it highlights some important aspects of this type of research. But before I do that, I just want to highlight when this paper was published. This was published in 2016, so almost 10 years ago. And there actually hasn't been really any significant characterization of other mutations beyond this paper until very recently. So in this paper, Tony's group collected a series of 78 melanoma patients. All of them were treated with a specific anti-PD-1 inhibitor, pembrolizumab. 42 of these patients had objective response to this drug. 15 ultimately had disease progression. And Tony decided to focus on 4 patients which had late acquired resistance because that was what he was interested in. So he defined this as those patients that had recurrence or progression in their disease 6 months or later after the initial tumor response. And so in these 4 patients that he studied, he did exome sequencing of the primary tumor and the resistant tumor, and we'll come back to that point in a minute. And he found 1 patient that had a JAK1 mutation, 1 patient who had a JAK2 mutation, 1 patient that had beta-2 a microglobulin mutation. And I'm not going to talk about what these mutations are, but these are all involved in interferon signaling and antigen presentation. And I think that, even though these numbers are pretty small, I think this paradigm that was established by this paper, where antigen presentation and interferon signaling exists to this day. And I think that probably one of the reasons that has been so successful in identifying mechanisms of acquired resistance was the fact that Tony sequenced a primary and resistant tumor and was able to compare those mutations that were present selectively in the resistant tumor. And we'll, again, talk about that in a minute. By the way, this is intended to be interactive, so if you have questions along the way, feel free to ask. Okay, so I think what Tony's paper suggested was that instead of taking a large number of patients, looking for associations with genetic mutations and seeing how they correlate to disease, so checkpoint inhibitor response, one can take another way, potentially with many fewer things, and identify these drivers of resistance in a more straightforward way. And so that was the impetus for our first foray into this. This was now published 5 years ago. I'll just very quickly take you through it. Again, this was our first patient that we studied. This is a patient of mine that had metastatic melanoma, had diffuse disease through the lymph nodes, a left adrenal lesion, tumors in the lung, and was treated with pembrolizumab. So very similar to what Tony had described in his series. And remarkably, all of these lesions disappeared. With the and you can see that here, this is lymph node highlighted in the circle, and that disappears after about a year of treatment. So all of these lesions disappeared with the exception of one, and that's here. The right adrenal mass circled here remained stable after a year of treatment. And so this patient underwent surgery to remove this lesion. And this created the opportunity to figure out what was unique about this adrenal mass compared to those tumors that actually did respond to treatment. So I wanted to show this. This is sort of, many of you in the data science world see figures like this, and I'm not used to seeing figures like this because I'm a molecular biologist. And you can see that each one of these names is a different gene that's mutated in the resistant tumor or the responding tumor. And you can see there's a huge number of mutations that are seen in these tumors. But most of these mutations are present equally in the resistant tumor and the responding tumor. So, for example, this cluster of mutations here, and even these clusters here along the diagonal, are present in equal proportion in the resistant tumor and the responding tumor. And so if you sort of subtract the sort of common mutations, you're left with a much more simple picture. And the picture is roughly 50 mutations that are unique to either the resistant tumor or the pre-treatment tumor. And among these 50 or so candidate mutations, there was only one mutation in a known tumor suppressor gene, and that was this mutation in FBOXW7. FBOXW7 is a E3 ubiquitin ligase, and the mutation that we saw in the resistant tumor, the R55C, was a known loss of function mutation in the FBOXW7. And so this led us to ask, was FBOXW7 required for response to immunotherapy or PD-1 inhibitor? So my lab is predominantly a wet lab, and so we explored this question using sort of standard molecular biology tools. Using CRISPR, we knocked out FBOXW7. You can see you can knock it out pretty well. And then we introduced these cells into syngeneic mice, and this is what we saw. So in black here, in the sort of dark black line here, that's the tumor, the wild type tumor that's introduced into these mice. When you treat it, this mouse with anti-PD-1, you see this dotted black line, so that the tumor is responding to the PD-1 inhibitor. And you can see that that effect is even more dramatic when you look at survival. So without treatment, the mice bearing the control tumor pass away. And then when you treat them with anti-PD-1, Many of them are cured, even with extended times. But what you see with the in the tumors that we've deleted FBOXW7 is that regardless of whether you treat them with anti-PD-1, they're resistant to the treatment, and they all pass away rather quickly. So then the question was, what is FBOXW7 doing? We decided to take a sort of transcriptional, a transcriptomic approach by characterizing by RNA sequencing differences in gene expression in the control cells or either of the two knockout cell lines that we derived. And the top gene set that emerged from this analysis was interferon signaling. In other words, when FBOXW7 was deleted, interferon signaling was turned off. You can see this is the signature here. Without anti-PD-1, you see some of these genes are expressed. They're increased in expression when we add anti-PD-1. These are the pluses here. And then in the FBOXW7 deleted tumors, there's lower baseline expression of these, and the addition of anti-PD-1 has a more dampened response compared to the control cells. And this is just validated here by quantitative PCR. Of course, this is just a mouse. So what about our patients? So we went back to the TCGA data set. We asked if you compared those tumors that had FBOXW7 mutations versus wild type, what were the differences? And here again, the number one gene set that was different between these two was the interferon alpha signaling response gene set.
Was there any difference in the immune infiltration of those groups?
There was. I won't go into it, but it's all in the paper. So how does FBOXW7 regulate type 1 interferon? I'm simplifying here, but there's two major pathways that regulate a type 1 interferon. One involves double-stranded DNA that activates a protein called STING. We'll talk about that in a minute. And then there's two pathways that converge on MAVs that respond to double-stranded RNA. And what we were able to show, and I'm not going to show you all those data today, is that FBOXW7 regulates the double-stranded DNA signaling. And this actually, in retrospect, should have come as no surprise because several years before our paper was published, another group had generated knockout mice lacking some of these double-stranded RNA sensors and found that they were more vulnerable to specifically RNA viruses. So they were unable to mount a sort of response to RNA viruses. So here's, so this led us to the question, could we activate this double-stranded RNA sensing pathway and promote response? And that's shown in this diagram. So again, the black line is the tumors that are wild-type tumors. But in this dotted line back down here, we've introduced MAVs genetically, and you can see that these tumors fail to grow, and this was dependent on an intact immune system. So I've taken you through that story, and that took us about 4 years to figure out. And then we said, well, let's do this on a bigger scale. And we assemble a cohort of patients that have acquired immunotherapy resistance. And as I said, the Tony Rivas paper was published in 2016, and until now, I don't believe there has been a cohort that has looked at this in a more comprehensive way. So we assembled a series of biopsies, like the patients that we were collecting biopsies from either had complete response to checkpoint inhibitors and had a recurrence, or like that first patient I told you about, had a complete response with the exception of a single resistant tumor. We collected 56 biopsies from 25 patients, and we did the same sort of analysis, exome sequencing, et cetera. And I'll just show you, this is a group of patients that had different kinds of treatment, different kinds of PD-1 or PD-L1 treatment, combination therapy, and even some clinical trials. I will just highlight that it's a sobering thought to think that this is years here. So you can see that in some cases, we collected biopsies almost 8 years after the initial biopsy was taken, which shows you some of the challenges of doing this sort of work, that you have to be patient to get those biopsies. And in fact, these biopsies were assembled from multiple institutions. So these are relatively hard to come by and rather rare. So in our cohort, we noticed 3 patients had mutations in this gene called SEC24C or SEC24. These are the 3 cases here. This case, by the way, is the case that Tony Ribas had not figured out. This is the 4th case that Tony Ribas's group had published in the first paper in New England Journal of Medicine, for which no candidate mutation had been identified. And in each one of these cases, the proportion of tumor cells expressing the mutant version of SEC24C or SEC24D increased in the resistant tumor compared to the pretreatment tumor. So what is SEC24C? Well, it turns out that SEC24C interacts with the double-stranded DNA sensor STING. Unlike the RIG-I or the MAVS pathway that I just described, STING is actually a molecule that is trafficked inside the cell. So it's typically present in the ER, and then it's trafficked to the Golgi complex, also called ERGIC, through a complex called COP2. And COP2 contains SEC24C and SEC24D, as well as SEC24D. And so previously had been shown, not in the context of immunotherapy, but in some other basic research, that SEC24 was essential for transporting STING to the ERGIC. So we did the same sort of studies that we showed you earlier. We deleted SEC24C in some melanoma cells. You can see that in the control cells, when you add a STING agonist, you increase the expression of type 1 interferon signaling, such as CXL10 or interferon beta-1 transcription. But when you delete SEC24C or SEC24D, I've been showing you the STING agonist is no longer able to activate these pathways. This, of course, just shows us that there's a requirement for SEC24C or SEC24D, but doesn't tell us that the mutations that we saw in our patients are sort of loss of function mutations. And so we did that experiment. So here we took some human cells, we deleted the endogenous SEC24C and replaced it with either wild type SEC24 or either of the mutants that we saw in our patients. And you can see that when you add a STING agonist, you induce interferon signaling, the wild type Sec24C, but not when the wild type Sec24C is replaced with one of these mutants that we saw in our patients. So this leads us to this kind of perhaps naive and overly simplistic model of acquired resistance to immunotherapy. I showed you that FBOXW7 impacts the double-stranded RNA sensing pathways through RIG-I stability. Sec24C and D impacts the STING pathway. And this points at a sort of a central role of these viral sensing pathways in regulating type 1 response and also response to any checkpoint inhibitors. But I want to take a step back and ask, what does that mean? Is this the answer, or are we just, where are we in understanding acquired resistance through genomic mediators? And so, as I told you, that we characterized 25 patients. 3 of our patients had Sec24C and D mutations. That leaves more than half of the patients for which we have yet to figure out if there is a genomic mediator of resistance to immunotherapy. And so my hope is that many of you who are in the data science world will help us figure this out. The data is freely available. We'd love to collaborate with you. I think there's a lot to be learned by deep mining of these data. That's beyond my pay grade for sure. So I will summarize. So I think that, in my view, even though this is somewhat a niche question of acquired resistance, it sometimes can inform us on pathways that are relevant in intrinsic resistance. Our studies have suggested a role for viral sensing pathways through either double-stranded RNA or double-stranded DNA pathways, and how we can exploit this information now to develop more precise ways of delivering immunotherapy remains still a challenge. We have abilities to activate so this agonists of double-stranded RNA and double-stranded DNA that exist and are in clinical trials, and how we exactly should be designing those studies to exploit this information, I think, is an area that needs some work. And as I mentioned, most patients that we've studied so far don't have a clear genomic mechanism of resistance. And that might mean that resistance is either epigenetic or perhaps related to microenvironmental influences in those patients, or that we've missed them. And this is where I love your help in figuring it out. So this is our lab. I wanted to just highlight that both of the studies I talked about were deep collaborations with Ellie and Dave. We worked with Tony, and we used samples from different institutions, including UCLA, Vanderbilt, and Dana-Farber. I will stop and happy to take questions.
So has anybody tried to profile a large enough cohort with DNA methylation intrinsic?
I don't think of anyone has.
But then and the obvious next.
It is the obvious question. Yeah. And I think, part of that might be related to just the paucity of these reagents. So, what I didn't mention is that when we embarked on this study, on the second study, we decided we'd do RNA sequencing. And I haven't shown you that data because we found that the samples, most of the samples in past QC. So there may be that may be part of the issue.
You see in the patients that the CRISPR mutation of the protein
SEC24.
SEC24, did you see a lack of accumulation of STING at the Golgi interstitial complex?
Yeah, we did,
immunofluorescence
we didn't look at that. It's a good question. We have not proven that that there is a effect. Other groups have shown that deletion of SEC24 leads to a blockage in the trafficking of things, and we didn't do that.
Great talk. I was wondering how much of the interferon response that you see when you take out FBX7 is related to the MYC increase that you also see on that gel. Do you think MYC is doing something else important, or is it known to be related to the interferon response or aggression of resistant state?
I think all we can say is that the double-stranded sensing is necessary for that interferon, but whether MYC is also part is necessary, we didn't look at that. I think the challenge is, suppressing MYC in a cell is usually toxic.
Right. I wonder if you overexpress MYC to copy the loss of FBX.
Yeah, so we don't.
Okay.
Yeah, it's definitely not sufficient. Whether it's necessary, we don't know.
But thanks so much.